Chemical transfection techniques. Here we will discuss different methods or some chemicals that can mediate DNA uptake by the mammalian cells. And first we will focus on the calcium phosphate mediated DNA transfer to the animal cells. The ability of the mammalian cells to take up the exogenous or foreign DNA was first time reported in 1962 when scientists used uncloned genomic DNA to transform animal cells. तो उस वक़्त जो एनिमल सेल यूज़ किया गया ट्रांसफॉर्मेशन के लिए तो वो एक ख़ास एंजाइम से डेफिशिएंट था या वो इस काबिल नहीं था कि वो एंजाइम एक्सप्रेस कर सके तो उस एंजाइम का नाम था HPRT एंड कॉमनली इट इज़ कॉल्ड एज हाइपोजेंथाइन गुआनिन फॉस्फोरिबोसाइल ट्रांसफेरेज और HPRT. Now, what is the function of this enzyme in the cells? This enzyme is very important for the recycling of nucleotides in the cell. For example, nucleotides, they are present in the DNA or RNA molecule. And this enzyme, HPRT, it has the ability to recycle such nucleotides. So here, sometime in the cells, de novo biosynthesis of the nucleotide, it is not required. Rather, pre-existing nucleotides, they are recycled. But the cell that was used to transform, it is lacking this ability, that is, ability to synthesize hypoxanthine guanine phosphoribosyl transferase or HPRT enzyme. And when the whole genomic uncloned DNA was used to transform the animal cell, then the wild type phenotype, that is, the ability to synthesize HPRT enzyme, it was restored. And later on we will also learn that how this ability can be used as a selectable marker. So at the time in early 1960s, scientists were not have any idea that how this genomic DNA it is being uptaken by the cells. Later experiments shown that actually calcium phosphate that is present in the medium, it form complexes with DNA, so that calcium phosphate DNA complexes are formed that are precipitated on the cell surface. After precipitation, such complexes were taken up by the cells by the process of endocytosis, and then the genomic DNA it was internalized, that later on transferred to the nucleus of the cell where it can integrate and express. Then with the passage of time, some scientists when used calcium phosphate method to increase the infectivity of the adenovirus, then this method was used as a general protocol for the transfection of the animal cells. This method, it can transform only those cells that are present or growing in the culture medium as monolayers or in the form of suspension, because when cells they are present in the form of aggregates or complex, then it is unable to transform such aggregated form of cells. Initially, it was found that calcium phosphate mediated DNA uptake, it is having low efficiency. That is, it is able to transform very limited number of cells. For example, ये देखा गया कि 1 से लेकर 2% तक जो सेल्स थे ये ट्रांसफॉर्म हो सकते थे. और इनमें से जो ज़्यादातर सेल्स थे जो ट्रांसफॉर्म हो भी जाते थे तो वो स्टेबली मेंटेन्ड नहीं रहते थे, यानी कि एक दो सेल डिवीजन के बाद उनकी ये जो ट्रांसफॉर्मेशन की जो एबिलिटी थी ये लूज हो जाती थी. देन अगेन the protocol was refined so that later on it was reported that by using calcium phosphate co-precipitation method, about 20% of the cells can be transformed. Then again, by using a specific type of buffered system that can control the abrupt changes in pH and that can slow the precipitation on the surface of the cell, the time for endocytosis was increased and the ability of the cells to take up exogenous DNA, it was enhanced 100-fold. But in case of calcium phosphate precipitation, particle size is very important, because if particle size is large, then transformation efficiency it can be influenced. And particle size, in turn, it can be controlled by the concentrations of the DNA, calcium, phosphate, and even the additives or components that we are going to add in the medium. They are also very important. So these are different factors that can influence the formation of particles and their sizes.